Welcome to the Global Virus Network's Forefront of Virology webinar series. I'm Dr. Maggie Bartlett, the Program Director for the GVN and your moderator today. We are so pleased to have Dr. Luigi Buonaguro. I practiced it and I'm so sorry. Buonaguro, who will present on molecular mimicry as a strategy for anti-cancer immunity. He is the director of the Innovative Immunological Models Lab at the National Cancer Institute in Naples and is focused on cancer immunotherapies and is really recognized for being one of the pioneers for the concept of molecular mimicry in cancer vaccine development. He has coordinated 2 major European consortia to develop anti-cancer vaccines and is leading the HepaVac 101 clinical trial. We are so pleased to have him enlighten us today. Please take it away.
So, can you hear me good? Can you hear me well?
Yes.
Okay. So, thank you, Maggie. Thank you, Ruby. Thank you, the GVN, for inviting me to have this webinar. I'm very happy and honored to be with you this evening. Well, this morning for you. Well, I have to just start saying that I was I was a part of the initiation of the GVN. I remember I was in Washington, D.C. I do not remember exactly when it was, but I was there among the ones who signed for the first time the beginning of this initiative. I remember we were at the Italian embassy to sign in Washington, D.C. to sign this. So I'm very happy to be here to present our data. So I will now share my screen. And okay, so as Dr. Bartlett introduced, my presentation is about the exploitation of the molecular mimicry for cancer vaccine development. So in order to have a more clear idea, I want to start just describing which kind of antigens we currently use in the field to prepare or to develop cancer vaccines. So we have mainly 2 classes of tumor antigens. The first one is named the tumor-associated antigens, the second tumor-specific antigens. So which is the major difference between these 2 types of antigens? The tumor-associated antigens are, generally speaking, wild-type cellular antigens, which are overexpressed in tumor cells. And as you may see in this cartoon, the expression of these tumor-associated antigens can be all over the body, although in a very low level of expression, but it is overexpressed in tumor cells. On the other side, tumor-specific antigens are antigens which are expressed only in the tumor or by tumor cells and not expressed by any other normal cell. So this means that obviously in terms of immunogenicity and specificity, tumor-specific antigens are the optimal antigens to be selected for developing a cancer vaccine, because there is no immunological tolerance, because they are basically non-self antigens, which are recognized by our immune system as non-self, and so they are a very good target for our immune system. On the other side, the tumor-associated antigens, because they are self-antigens overexpressed in tumors, they may suffer from some levels of immunological tolerance. So they would be not the first choice to make a cancer vaccine. If we consider what they are, so tumor-associated antigens are cancer germlines, or from differentiation, or overexpressed in tumors. But as I said, these are wild-type self-antigens overexpressed in tumors. On the other side, tumor-specific antigens are either viral antigens, for example, in HPV or HBV, so papillomavirus or hepatitis B virus associated cancers, or they are the outcome of mutations like single nucleotide variations, insertion or deletions or fusion, and so on. So, as I said, if you would bet, you would say, okay, I want to make a cancer vaccine with the tumor-specific antigen. They are not self, they are new, they are not seen by our immune system as something already present in our body, so there is no immunological tolerance. But there is a but, there is a big but. Tumor-specific antigens are mostly individual. So this means that for each individual patient, we must make all the analysis, the sequencing, the prioritization of these new antigens, the validation, the manufacturing of the vaccine, this for each individual patient, because these tumor-specific antigens are mostly individual for each patient. And even in the same patient, if after 6 months, after 1 year, the tumor would relapse, very likely the new tumor, the relapsing tumor, will not have the same mutations, so the same neoantigens to be used for vaccine. So all this process needs to be done all over again. And right now, it is estimated that for each individual vaccine, for each individual patient, this would cost about $400K USD. So you may understand that this is very complicated, very cumbersome, very difficult to be produced, to be used as a mass vaccination strategy. Obviously, we would like to have some mutated tumor-specific antigen, some mutated neoantigen shared among cancers, shared among patients, in order to produce an off-the-shelf vaccine. Unfortunately, this is not the case, as we have recently described. Indeed, we have analyzed the most frequent mutations which are identified in the cancers. So we look at the TCGA, where we took the most frequent mutations which occur in about 56% of all reported cases in all the TCGA. And we decided to look for neoantigen prediction from these mutations, neoantigen prediction in 50 HLA, covering the 3 major alleles, A, B, and C, and in this way covering mostly all the human population on Earth. Well, the point is that at the end of the day, if we look at the single nucleotide variations or at the insertion deletion variation, you may see that the number of predicted shared tumor neoantigens which could be used for off-the-shelf vaccine development are very, very limited. And the point is also that they are associated with the HLA haplotype, which are unfortunately the less present, the less represented in the human population. So this means that if we consider the different tumors where these mutations, as I said, the ones that are found, that are deposited at the TCGA, we could eventually treat these percentages of these different tumors. For example, 25% of the stomach cancers or 13% of the pancreas tumors. But the point is that only in some patients across the globe could benefit from this off-the-shelf vaccine. So again, at the end of the day, this strategy, so generating, developing cancer vaccine, off-the-shelf cancer vaccine, based on shared neoantigens, is a strategy which can be used, but could be beneficial only for a very minority of cancer patients. So why do we have mutations, so frequent mutations? Let's consider the P53 mutation, the BRAF mutation, KRAS mutations, which are very much predominant in many tumors. But why these mutations do not give rise to targetable neoantigens? Probably because these have been selected through cancer development. So this means that all the mutations which are easily seen, recognized, and targeted by the immune system in the very early stages of tumor growth, they are gone. So only the mutations which do not give rise to mutant peptides, mutant antigens, so they are not seen by our immune system, well, these are hidden, so they can grow, they can proliferate, and so our immune system is unable to clear these tumors, and that's why at the end of the day we find those tumors with those mutations which are not seen by our immune system. So although tumor-associated antigens, which are, again, wild-type overexpressed antigens, may suffer from immunological tolerance, so they may be poorly immunogenic, they represent at least for us, our main strategy to be followed in order to be pursued in order to develop a cancer vaccine. But, as I said, there are some problems because there is a central immunotolerance and therefore low immunogenicity. So we need to solve this problem. And so which is the strategy that we adopt in our lab at our institute? So first of all, we identify specific tumor-associated antigens, so the ones which are mostly expressed by cancers, and then we improve, or we look for improvement in immunogenicity of the tumor-associated antigens, namely by molecular mimicry. So whatever I'm going to present has been published, and I just am showing here two recent reviews which we published in this field. So the T-cell recognition of epitopes, of antigens, occurs in what I like to call the ménage à trois. Indeed, the epitope, the antigen, can be recognized by the T-cell receptor of T-cells only if it is presented by the MHC class I molecules. And in this case, I'm showing, for example, the HLA-A0201. In this conformation, in this presentation, the epitope is showing to the TCR only some of the amino acid residues along the sequence. Please remember that for the MHC class I, the length of the antigen is between 8 to 10 amino acids. So, as you may see, only some of the residues, namely the ones which are in the core position, are those which are in contact with the alpha and beta chain of the TCR. So if we want to find something similar, the similarity must reside in these residues along the antigen sequence. But in order to take advantage of the molecular mimicry, we must consider that our T cells are able to cross-react. This means indeed that T cell receptors can recognize at least 1 million of different peptides bound to the MHC. And this is absolutely necessary because it has been considered that we can be exposed to about 10 to the 15 potential epitopes. But our T cells are only 10 to 12. And actually, only 10 to 8 are naive. So they can be newly activated by a new antigen. So the initial paradigm stating that each TCR could recognize only one antigen obviously is not possible, because if this would have been true, there would have been about 10 to the 7, so about 10 million antigens which could not be recognized by our antigen, by our T cells. So this means that each TCR can recognize about 1 million antigen, obviously very similar antigens, presented by a molecule of the MHC. And indeed, a few years ago, it has been postulated that no one is naive. So meaning that basically in our body, there are T cells which even we have not been exposed to a specific antigen, what we already have in our body, circulating in our body, is actually already able to recognize something completely new that we would eventually encounter for the first time. Because as you may see, the TCR can recognize something which is similar, molecular mimicry. We can have alternative recognition. We can have also the same T cell recognizing two different peptides on two different MHC. So again, this is just to state that because of this degeneration, I would say degeneration or cross-reactivity of our T cells against a broad spectrum of similar antigens presented by the MHC, we can leverage on homology, on similarity, on the molecular mimicry to improve the efficacy of cancer vaccines against tumor-associated antigens, which are wild-type, overexpressed self-antigen, but they are shared among patients. So in the reviews that I previously shown, there is a very complete list of homologies between tumor-associated antigens and viral antigens. And here I show what we have contributed in the literature in the last few years. And again, instead, this is the example of homologies between tumor-associated antigens and bacterial antigens. And again, here is the list of what we have contributed in the recent years. So you see, there are few examples of these homology between microbes, meaning viruses or bacteria, and tumor antigen. And this is based on a sequence alignment. So let's start with some specific cases that we have described recently in the literature. For example, the HIV. So as you know very well, I assume that among the people in this webinar, there are many people involved in HIV field. There are the incidence rates of AIDS-defining cancers are significantly greater in people living with HIV/AIDS than in the general population. But non-AIDS-defining cancers show variable standardized incidence ratios compared to the general population. And there are some of these non-AIDS defining cancers, namely breast, prostate, colon cancer, which actually are reduced compared to the HIV negative people. So we asked, could this be due to a molecular mimicry between antigens derived from HIV and tumor antigen? Well, the answer is yes. Here, I reported some of the antigens, some of the tumor-associated antigens overexpressed in breast, prostate, colon, which show a very high homology. Please look at the aligned sequences, very high homology with the epitopes antigen derived from HIV proteins. And taking into considerations about 5,000 HIV isolates, we showed that this homology is not with a single variant, a single species of HIV, but they are conserved among about 5,000 HIV isolates. And this is just to show you that indeed, if we look at the conformation of the paired tumor-associated antigens and HIV epitope, actually we see that they are extremely similar in terms of conformation, in terms of contact with HLA, the green part of these structures. And we confirm that in this is not only a speculation, is not only a sequence homology, but indeed, if we look at the HIV positive, when these individuals living with HIV are exposed in vitro with the tumor-associated antigen, here you can see that only the HIV positive individual show a very significant positivity in ELISpot assay compared to HIV negative. So this is the first demonstration that indeed individuals which are infected and live with HIV have T cells circulating in their body which have been elicited by HIV protein, HIV epitope, but these T cells are able to cross-react and recognize also tumor-associated antigens. And this could be one of the explanations why non-AIDS defining cancer can have an incidence in HIV people lower than not or HIV negative individuals. Then we looked into the molecular mimicry or similarity or homology between tumor-associated antigen and the microbiota antigen. And for this, we took into consideration bacteria with the family within the gene of Firmicutes and Bacteroidetes, which altogether they represent about 70% of the microbiota in our body, intestinal microbiota. And this is just to show how many antigens are indeed showing a very high homology between these bacteria, this microbiota, and tumor-associated antigens, both in HLA 0201 and 2402, which altogether represent about 50% of the Western population. And this is just to show you what means, what we are considering homology. The green are the different residues along the sequence. The dots indicate completely homology, identity between the tumor-associated antigen in red and all the different antigens derived from different microbiota populations. And just to show you one example of this homology, this is the MAGE1, which is one of the most important tumor-associated antigen overexpressed in several tumors across individuals, and one of these antigens or epitopes derived from microbiota strains. You see, this is always the molecule, the peptide in the conformation when it's bound to the MHC class 1 and presented to the T cell, and as you may see in circle, identical residues. And this is just to show how the epitope protrudes from the MHC when it is in the MHC. But even more important, I want to show you the footprint of these two antigens on the inner phase of the alpha and beta chain of the TCR. Please consider the red areas. The red areas are the footprint of the MAGE1 on the left and microbiota-derived epitope on the right. They are basically identical. So this means that if there is a T cell in our body which has been exposed to this microbiota antigen, it will recognize for sure, 100%, it will recognize the tumor-associated antigen, the MAGE1 tumor-associated antigen presented by a tumor. And indeed, we also confirmed that, again, this homology is not only specific to a single strain, but there is a very high conservation of these sequences. We showed that both healthy individual and cancer patient can cross-react with the tumor-associated antigen and the homologous microbe-derived antigen, MOA. And finally, we showed also that if we vaccinate in vitro, ex vivo T cells with the microbe-derived antigen, and then we challenge these activated T cells on cells, cancer cells, expressing the homologous tumor-associated antigen, we see a CTL, a cytotoxic activity. So, looking at these data, we thought, do we have a vaccine? Can we have a vaccine, a cancer vaccine based on microbe vaccines? Or can we exploit what we already have? So we looked for homologies in the proteins presented or administered with the mandatory preventive vaccines for infectious diseases. So the mandatory vaccines, which every one of us has been exposed, has been administered in his or her life. Well, the data are very striking. So if we consider, let's consider the combination of vaccines that are used all over the world. So the MMR, the hexavalent, and in total, you see how many antigens, predicted antigens in these different HLA are potentially presented to the immune system when we are vaccinated for these infectious diseases. Obviously, this must be confirmed in vivo in individuals because, as you know, these vaccine formulations are prepared, are manufactured in order to elicit mostly the B cell response, so the antibodies response. So we must confirm now in vivo in individuals how many and how strong T cell responses we have after a vaccination in pediatric age. But potentially, we have shown that potentially there are a very good number of antigens which are expressed by preventive vaccines, which could already represent, at least in theory, a preventive vaccine for cancer. And this would be a great news. And this is, again, one example. So this is one of the tumor-associated antigen, which shows a very high homology in terms of sequence, conformation, contact with the TCR, with an epitope derived from the diphtheria vaccine. And here, this is just a recapitulation, just to show the potential effect of a preventive vaccine, because on the left are listed the tumor-associated antigens, on the right, the name of the cancers, and here the numbers indicate so how many tumors each of these tumor-associated antigens could prevent, or how many tumor-associated antigens can be preventing each tumor when or after the preventive vaccines. So I want to conclude this analysis looking at the SARS-CoV-2, at the molecular mimicry of SARS-CoV-2 and tumor-associated antigens. This is a paper that we published last year. So basically, we took into consideration the proteins included in the vaccine. And actually, in the paper, if you look into the paper, you will see also the proteins which are outside of the vaccine. But today, I want to focus on the proteins which are included in the vaccine, so in the spike of the SARS-CoV-2. Well, you may see very clearly that there are several examples of epitopes from the spike included in the SARS vaccine, which show a very high homology with the tumor-associated antigens, which are overexpressed in the tumors listed on the right side. Again, this is to show what we mean as homology. So not only the similarity in the linear sequence, not only the similarity of the conformation, but mostly the footprint that the two epitopes, the two antigens, have on the inner face of the TCR. So again, these are epitopes which are very similar in the way they are seen by our TCR. So this implies that when we have been vaccinated, we have the vaccine has generated not only the antibodies, the neutralizing antibodies, but also T cells, which now are able or potentially able to recognize tumor-associated antigen. And this is just an example. On top, I show just the single individual cross-reacting with the matching or the matched SARS antigen and the tumor-associated antigen. And below is instead the collection of multiple reaction from different individuals, where we show not only the reaction against the viral or the tumor-associated antigen, but also the cross-reaction in the panel C. Well, Probably you are aware that just a few days ago, it has been published a very interesting paper in Nature, where they have retrospectively observed, and this is from the MD Anderson, that patients, cancer patients, namely lung cancer patient and melanoma patient, when they have been vaccinated with the COVID vaccine, the mRNA COVID vaccine, within 100 days before starting the immunotherapy, the immunocheckpoint inhibitor therapy, well, as you may clearly see, the overall survival in the vaccinated patient, the COVID vaccinated patient, the overall survival is significantly much better than those who have not been vaccinated before the beginning of the checkpoint inhibitors. In the conclusion or in the interpretation of the authors, they state that the mRNA anti-SARS vaccine has been able to elicit a potent immune modulation, which is able to make the tumors more sensible to the immunocheckpoint inhibitor effect. We believe, obviously, what they have proposed for sure is one of the mechanisms. We believe that in association with this interpretation, there is also the interpretation of the cross-reactivity between antigen from the vaccine, from the SARS vaccine, and tumor antigen expressed by these tumors. And we hope to have the opportunity to make a joint effort with the colleagues at the MD Anderson to indeed show that these patients with the much better overall survival have a greater or higher frequency of cross-reactive T cells. So this is the mechanism that we think that occurs. So basically, dendritic cells which are presenting antigens derived from microorganisms elicit T cells. These T cells are able to recognize tumor-associated antigens very similar to the microbe-derived antigen. And so this means that now the T cells elicited by the microbe antigens now are able to cross-react and have a very strong effect on T cells, on tumor cells, especially if this memory against the microbe antigens has been established before the growth of tumors. Because in this way, the primed immune system can act in the very early stage of the disease, of the tumor disease, even when the tumor has not been already embedded in the tumor microenvironment, which is highly immune suppressive. So can we exploit molecular mimicry for the cancer vaccine? Yes. That's what we are pursuing right now. We have selected proteins overexpressed in hepatocellular carcinoma, identified epitopes from these tumor-associated antigens with a high homology with the viral-derived antigens. And again, these are the paired antigens. I want just to show again in yellow, you see the contact points with the HLA, and the magenta and the blue are instead the contact points with the alpha and beta chain of the TCR, the T cell receptor. And you can see that if you look at all the different pairs, they are basically indistinguishable. We show that indeed in HCC patient, we find T cell able to cross-react with the tumor-associated antigen and the viral antigen with a high homology. So based on this concept, we have developed, designed a cancer vaccine with a mix of 10 antigen, some of which are derived from the virus, from viruses with homology with the tumor-associated antigen, hoping to be able to elicit a cross-reactive T cell. And we plan to start the phase 1 clinical trial in the first quarter of 2026. I want to finish with this picture because given, as I showed you before, that, for example, the MAGE tumor-associated antigen is overexpressed in all the tumors that I have highlighted in this picture, in this cartoon. We can propose that if we develop a cancer vaccine with a microbe-derived antigen similar to the MAGE, basically we are developing one vaccine for many tumors. So not only an off-the-shelf vaccine for many patients with the same cancer, but one vaccine for many patients with many different cancers. So this is the completely opposite of the highly personalized cancer vaccine that I showed you at the beginning. And this is just to show the pipeline that we follow. So we identified tumor-associated antigen shared among tumors. We look for homology between the tumor-associated antigens and the microbe-derived antigen. We confirm that indeed there is an immune recognition by T cells, and then we develop a vaccine for phase 1 clinical trial. So I hope that I convince you that there are several examples of sequence and structural homology between the tumor-associated antigen and the microbe-derived antigens, and that indeed the microbe-derived antigens represent a novel class of non-self antigens, highly immunogenic antigens to be used in cancer vaccine development. Indeed, these are non-self, highly immunogenic. They elicit potent T-cell response, which is able to cross-react with the real tumor-associated antigen presented by tumor cells. So we believe that in the next generation cancer vaccine, microbe-derived antigens should be always included in these formulations. So I want to acknowledge all the collaborations at the Institute and all over Europe and the world to these studies. But most of all, I want to thank the collaborators in my lab, which actually are those who really do the job while I make all the paperwork or grant writing. So thanks to them, because without them, all this story could not be told to all of you. Thank you very much.
Thank you so much for an enlightening presentation. I have so many questions, but I'm going to first address the ones in the chat for you. We have one from David Ostroff. For the clinical trial, are the peptides mixed with adjuvant and injected intravenously?
Okay. Obviously, yes, peptides are mixed with adjuvants because otherwise peptides are not immunogenic. And they are administered intradermally, not intravenously, intradermally.
Mm.
Because as many of you probably know, the derma is the place where we have the highest concentration of resident dendritic cells, namely Langerhans cells. So we want to hit the Langerhans cells have the Langerhans cells then carrying or taking the antigens to the to the node in order to prime and start all the T-cell reaction.
That's fascinating. Stuart Isaac says, wonderful talk. I learned a lot. For the recent data on COVID vaccine and cancer, has anyone looked at a similar cohort vaccinated with protein-based COVID vaccines, not mRNA-based?
It's a matter of availability of patients, or these are not patients. I'm sorry, of individuals. At least in Italy, I think that 99% of people have been vaccinated with mRNA vaccine. So I think that we would have at least in Italy, very much a hard time to find individuals vaccinated with a peptide-based vaccine.
That makes sense. What about between those who had SARS-CoV-2 infection and those who were never infected or uninfected?
Sorry, I didn't get the question. I'm sorry.
So it's a follow-up for the recent data on the COVID vaccine and cancer. Yeah. If we're seeing any differences between, I suppose, those who were vaccinated, those who were never vaccinated after they got an infection, and if there are differences in rates in cancer.
Well, this is too early. It's too early to make this assumption. We have some preliminary, very preliminary observation in our lab that there is some correlation between SARS, not SARS-CoV-2, SARS, and cancer and cancer incidence. But this is too early to speculate now.
In your opinion, what would be the biggest hurdle in moving these molecular mimicry-based vaccines from the preclinical studies to clinical trials? You're already well on your way, but are there hurdles that you foresee or that you're facing?
Well, obviously, one of the questions that I always are asked during my presentations is, are you scared about possible autoimmune response? Okay, so my answer is always the same. So first of all, in the last probably 20 years, 25 years, thousands, well, I don't know thousands, but for sure hundreds of clinical trials have been performed using tumor-associated antigens. So exactly the same antigen, the same self-antigen, which are presented by our tumors. And to my knowledge, none of these cancer vaccine trials have been stopped because of serious adverse events in terms of autoimmunity. So I don't know why we should have these with these antigens. But then there is also another observation. These antigens are those which we are exposed to every time we get infected. Obviously, we know that there are cases of autoimmunity post-infection, but these should be taken into consideration as, I would say, a kind of a background. So the point always is always a balance. So what do we have to look for? The benefit or the possible adverse events? For sure, we could have some adverse events, but I don't think that they would be more frequent than the vaccines based on tumor-associated antigens.
Thank you. I think that is really important to keep in mind, is the nuance of different things and what's been identified in other vaccine approaches. I wonder if we might, because AI has become ever so dominant in our scientific conversations, how you think that might influence epitope prediction or the design of these vaccines over the next 5 to 10 years.
A lot, because these are all big data. So AI will be, for sure, will foster all our studies, because what I have showed to you has been done by hands or by eyes, I would say. So the more all the bioinformatics tools and artificial intelligence tool will be developed, the more we will find additional evidences about the molecular mimicry.
That's very helpful. Do you see any particular technologies or platforms that might be useful in transforming the mimicry-based cancer vaccines that you've been spearheading? Or
probably I don't get the real point of the question. I'm sorry.
I guess I'm looking for if there are potential new technologies or platforms that'll help accelerate the work that you're doing, or if we're
well, as I said just a few seconds ago, for sure the artificial intelligence will speed up all the discovery. That's for sure. That's for sure. In terms of vaccine platform, if the question is about I don't know, mRNA based or peptide based or whatever based, well, that's something which we cannot predict. The point is that the mRNA vaccine is a big obstacle, because the vaccine by itself is not very expensive, but there are a lot of royalties to be taken into considerations for the final formulation. So that's really a problem in terms of feasibility, in terms of applicability in clinical trials.
That's very helpful. I wonder if you might talk about what we should anticipate looking out for over the next year in this space.
Well,
without giving away any of your pre-publication data.
Well, as I said, we are starting this new, we are planning to start this new clinical trial in next year, early next year. So we hope to have, to be able to have a fast enrollment of patients in order to have results or preliminary results by the, I mean, in 9-12 months. So that would be the first point. Then there are several lines of research in the lab, which will give us for sure, and which are giving us many other insights of all these mechanism, or about the mechanism of the protection or cross protection induced by or elicited by molecular. So yes, stay tuned. Yes, for sure. Yes.
Yeah, we certainly will. Thank you so much for this truly inspiring talk. I want to, I do see one more question and I'll allow it. Franco is asking, is there any trial on administering just the COVID vaccine adjuvants to verify the effects of auto vaccination with their cancer-associated tumor antigens?
The COVID vaccine adjuvant?
Yes, I'm not.
Well, if we consider, if you look at the mRNA vaccines, they do not have adjuvants, because the mRNA by itself is an adjuvant. So I don't really get the point of this question. Probably I did not understand correctly the meaning of the question. So if the question is about using the adjuvant which has been used in the COVID vaccine for developing an anti-cancer vaccine, if this is the question, the answer is this is not applicable because, as I said, in the mRNA-based vaccine, there is no added adjuvant because itself, the RNA is an adjuvant.
Yeah, I would say Stuart Isaacs is adding for us the lipid nanoparticle without mRNA could be considered that. Yeah.
So the answer, so the question is, have we formulated the peptides in the nanoparticles which are used to deliver the mRNA? That's the question.
Yes.
Well, yes, we have been doing some experiments in vivo, in vitro, ex vivo. We don't see a real improvement, so I don't think that this is necessary. And the nanoparticles used by the companies for making the mRNA vaccine are under IP protection. If you want to use those nanoparticles for your vaccine, you may be asked to pay around 2 to 20 million only for using those nanoparticles which have been proven to be effective in clinical trial. So personally, I would not go after that.
That's very helpful. I want to remind everybody that this is a CME eligible lecture. The documentation for that is in the chat. Thank you, Ruby, so much. Thank you, Dr. Buonaguro, for your fantastic talk. We look forward to seeing you all next time on this webinar series of the Forefront of Virology. And thank you again for everybody who submitted questions and for our speaker today.